Welcome to Loops and Loops, your portal to the captivating world of biology. Biology, the study of living organisms and their interactions with the environment, offers insights into the wonders of life itself. Join us as we embark on an enlightening journey through the realm of biology, exploring the diversity of life forms, unraveling the mysteries of genetics, and delving into the intricate ecosystems that sustain life on Earth. Whether you're a biology enthusiast, a curious learner, or simply intrigued by the complexities of the natural world, you're in for an immersive experience. So tighten your loops, prepare to delve into the wonders of life, and let's explore the fascinating world of biology together. Biology is the scientific study of living organisms and their interactions with each other and their environment. It encompasses a wide range of topics, from the structure and function of cells to the behavior and evolution of organisms. Biology is crucial because it helps us understand life in all its forms, from the tiniest microorganism to complex ecosystems. It allows us to explore the processes that govern life, including growth, reproduction, and adaptation. By studying biology, we gain insights into how living organisms interact with each other and their environment, which can inform fields such as medicine, agriculture, and conservation. The basic units of life are cells, which are the smallest structural and functional units of living organisms. Cells are capable of carrying out the processes necessary for life, such as metabolism, growth, and reproduction. DNA is a molecule that carries the genetic instructions for the development, functioning, growth, and reproduction of all known living organisms. DNA encodes genetic information through a specific sequence of nucleotides, which are the building blocks of DNA. These sequences contain the instructions for building proteins, which are essential for the structure and function of cells. RNA is a molecule that plays a crucial role in gene expression. It acts as a messenger between DNA and the protein synthesis machinery in cells, helping to translate the genetic code into functional proteins. Genes are segments of DNA that contain the instructions for producing specific proteins. They determine traits such as eye color, hair texture, and susceptibility to certain diseases. Genomics is the study of an organism's entire genome, which includes all of its genes and non-coding DNA sequences. It has revolutionized biology by providing insights into the structure, function, and evolution of genomes. Chromosomes are thread-like structures found in the nucleus of cells that contain the genetic material or DNA. They contribute to inheritance by carrying the genes that determine an organism's traits from one generation to the next. Epigenetics is the study of changes in gene expression that are not caused by changes in the DNA sequence itself. It influences gene expression by modifying the structure of DNA and its associated proteins, affecting how genes are turned on or off. Cell biology is the study of cells, their structure, function, and behavior. It is important in understanding life because cells are the basic building blocks of all living organisms and are responsible for carrying out the processes necessary for life. There are many different types of cells, each with specific functions and characteristics. For example, nerve cells transmit electrical signals, muscle cells contract to produce movement, and red blood cells transport oxygen throughout the body. Cell division is the process by which cells divide to produce new cells. It is essential for growth, repair, and reproduction in multicellular organisms. Metabolism is the set of chemical reactions that occur in living organisms to maintain life. It involves processes such as the conversion of food into energy and the synthesis of biomolecules needed for cell function. Enzymes are proteins that catalyze biochemical reactions in cells by lowering the activation energy required for the reaction to occur. They play a crucial role in regulating the rate of metabolic pathways and other cellular processes. Photosynthesis is the process by which plants, algae, and some bacteria convert sunlight into chemical energy in the form of glucose. It involves the absorption of light by chlorophyll molecules in chloroplasts, which drives the synthesis of organic molecules from carbon dioxide and water. Cellular respiration is the process by which cells convert glucose and oxygen into energy, carbon dioxide, and water. It occurs in multiple stages, including glycolysis, the citric acid cycle, and oxidative phosphorylation. Through these processes, cells generate adenosine triphosphate, which serves as the primary energy source for cellular activities. The main branches of biology cover a wide range of topics. These include genetics, the study of heredity and variation; ecology, which examines the interactions between organisms and their environment; physiology, the study of how living organisms function; and microbiology, the study of microscopic organisms like bacteria and viruses, among others. Plants and animals are two distinct kingdoms within the classification system of living organisms. Plants typically produce their food through photosynthesis, have cell walls made of cellulose, and are generally stationary. In contrast, animals are heterotrophic, meaning they obtain their food by consuming other organisms, lack cell walls, and are typically capable of movement. Living organisms obtain energy through various metabolic processes. Photosynthesis is one such process occurring in plants, algae, and some bacteria, where light energy is converted into chemical energy stored in glucose. Cellular respiration, on the other hand, is the process by which cells break down glucose to release energy, which is then used to power various cellular activities. Cells are the basic structural and functional units of life. They vary in size, shape, and function, but share common features such as a cell membrane, genetic material, and the ability to carry out essential life processes like metabolism, growth, and reproduction. Cells reproduce through processes such as mitosis and meiosis. Mitosis is a form of cell division that produces two genetically identical daughter cells, essential for growth, repair, and asexual reproduction. Meiosis, on the other hand, is a specialized form of cell division that produces gametes with half the number of chromosomes necessary for sexual reproduction. DNA determines our traits through the process of gene expression. Genes, which are specific segments of DNA, contain the instructions for building proteins, the molecular machines that perform various functions in the body. Different combinations of genes lead to variations in traits like eye color, height, and susceptibility to diseases. Ecology is the study of how organisms interact with each other and their environment. It examines the distribution and abundance of organisms, the flow of energy and nutrients through ecosystems, and the factors that influence these patterns. Ecology plays a crucial role in understanding and managing ecosystems, conservation efforts, and addressing environmental challenges such as climate change and habitat loss. Organisms interact with their environment in various ways to meet their needs for survival, growth, and reproduction. These interactions include obtaining food, water, shelter, and mates, as well as avoiding predators and competitors. Organisms may also modify their environment through activities like burrowing, nesting, and building. Biodiversity, or biological diversity, refers to the variety of life forms on Earth, including species diversity, genetic diversity, and ecosystem diversity. Biodiversity is essential for ecosystem stability, resilience, and functioning. It provides ecosystem services such as pollination, nutrient cycling, and climate regulation, which are vital for human well-being. Ecosystems are communities of living organisms, which is biotic components interacting with their physical environment, which is abiotic components such as soil, water, and climate. They range from small-scale habitats like ponds and forests to large-scale biomes like deserts and oceans. Ecosystems are characterized by energy flow and nutrient cycling among organisms. Food chains and food webs illustrate the flow of energy and nutrients. Through ecosystems, in a food chain, energy is transferred from one organism to another as they consume each other. A food web is a more complex network of interconnected food chains, showing multiple feeding relationships within an ecosystem. Producers, such as plants and algae, are organisms that produce their food through photosynthesis, converting light energy into chemical energy, stored in glucose. Consumers, including herbivores, carnivores, and omnivores, obtain their energy by consuming other organisms. Symbiotic relationships are close and long-term interactions between different species. Examples include mutualism, where both species benefit; commensalism, where one species benefits while the other is unaffected; and parasitism, where one species benefits at the expense of the other. Decomposers such as bacteria, fungi, and detritivores break down dead organic matter into simpler substances and recycle nutrients back into the ecosystem. They play a crucial role in nutrient cycling and the decomposition of organic waste. Biomes are large-scale ecological regions characterized by distinct climate, vegetation, and animal life. Examples include tropical rainforests, deserts, grasslands, and tundras. Each biome has unique adaptations and species composition adapted to its specific environmental conditions. Organisms adapt to their environment through evolutionary processes such as natural selection, genetic variation, and behavioral adaptations. These adaptations may include physical traits, physiological processes, and behavioral strategies that enhance an organism's survival and reproduction in its habitat. The water cycle, or hydrological cycle, describes the continuous movement of water between the atmosphere, land, and oceans. It includes processes such as evaporation, condensation, precipitation, infiltration, runoff, and transpiration. The carbon cycle describes the movement of carbon atoms through various reservoirs, including the atmosphere, oceans, soil, plants, and animals. It involves processes such as photosynthesis, respiration, decomposition, combustion, and fossil fuel combustion. Human activities, such as deforestation, urbanization, pollution, and greenhouse gas emissions, have significant impacts on the environment, including habitat destruction, loss of biodiversity, air and water pollution, and climate change. Deforestation, the clearing of forests for agriculture, logging, and urban development, leads to habitat loss, soil erosion, loss of biodiversity, and disruption of ecosystem services. It contributes to climate change by releasing carbon stored in trees and reducing the Earth's capacity to absorb carbon dioxide. Climate change refers to long-term changes in temperature, precipitation patterns, and weather extremes caused by human activities, primarily the burning of fossil fuels and deforestation. It has far-reaching impacts on ecosystems, weather patterns, sea levels, agriculture, and human health. The greenhouse effect is a natural process whereby certain gases in the Earth's atmosphere, such as carbon dioxide, water vapor, and methane, trap heat from the sun and warm the planet's surface. However, human activities have intensified this effect by increasing the concentration of greenhouse gases, leading to global warming and climate change. Renewable energy sources are natural sources of energy that are replenished over time, such as sunlight, wind, water, biomass, and geothermal heat, unlike fossil fuels, which are finite and renewable energy sources, can be continuously replenished and are considered more sustainable and environmentally friendly alternatives for meeting energy needs. Animal adaptations are special features or behaviours that help organisms survive and thrive in their environments. Examples include camouflage, mimicry, protective coloration, structural adaptations such as wings for flight or fins for swimming, behavioural adaptations such as hibernation or migration, and physiological adaptations such as the ability to regulate body temperature. Herbivores are animals that primarily eat plants. The carnivores are animals that primarily eat other animals, and omnivores are animals that eat both plants and animals. These classifications are based on an animal's diet and feeding habits. Animal behaviour refers to the actions, reactions, and interactions of animals in their environment. It encompasses a wide range of activities, including feeding, mating, parenting, social interactions, migration, and territorial defence. Migration is the seasonal movement of animals from one place to another, typically for purposes such as finding food, breeding, or escaping harsh weather conditions. Animals use various cues, such as changes in day length, temperature, and food availability, to navigate their migration routes. Insects and spiders are both arthropods, but they belong to different classes. Insects have 3 body segments like head, thorax, abdomen, 6 legs, and typically 2 pairs of wings. Spiders, on the other hand, have 2 body segments like cephalothorax, abdomen, 8 legs, and no wings. Mammals are warm-blooded vertebrates, characterized by features such as mammary glands for milk production, hair or fur covering the body, a 4-chambered heart, and the ability to regulate body temperature internally. Birds fly by using their wings to generate lift and thrust. Their unique anatomy, including lightweight bones, powerful chest muscles, and aerodynamic feathers, enables them to achieve powered flight. Birds also have a highly efficient respiratory system and a keen sense of navigation. Reptiles and amphibians are both cold-blooded vertebrates, but they belong to different classes. Reptiles have dry, scaly skin, lay eggs with leathery shells, and breathe through lungs. Amphibians, on the other hand, have moist, permeable skin, lay eggs in water or moist environments, and undergo metamorphosis from aquatic larvae to terrestrial adults. The life cycle of a butterfly consists of 4 stages. Egg, larva, which is caterpillar, pupa, which is chrysalis, and adult, which is butterfly. After hatching from an egg, the larva feeds voraciously, molts several times, and eventually forms a pupa. Inside the pupa, the larva undergoes metamorphosis and transforms into an adult butterfly. Plants reproduce through both sexual and asexual methods. Sexual reproduction involves the fusion of male and female gametes, which is pollen and ovule, to form a seed, which develops into a new plant. Asexual reproduction includes methods such as vegetative propagation, where new plants grow from existing plant parts like stems, roots, or leaves. The flower is the reproductive structure of a plant and typically consists of 4 main parts. Sepals, which is used for a protective outermost layer, petals, which is coloured structures that attract pollinators, stamens, which is male reproductive organs that produce pollen, and pistils, which is female reproductive organs that contain ovules. Pollination is the transfer of pollen grains from the male reproductive organs, which is anthers to the female reproductive organs, which is stigma of flowers, leading to fertilization and seed production. Pollination can occur through various mechanisms, including wind, water, insects, birds, and other animals. Seeds germinate when environmental conditions such as moisture, warmth, and oxygen are favourable for growth. During germination, the seed absorbs water, swells, and breaks open. The embryo inside the seed begins to grow, sending out roots to absorb water and nutrients from the soil and shoots to emerge above the ground and start photosynthesizing. Plants adapt to different environments through various mechanisms to ensure their survival and reproduction. These adaptations include modifications in their physical structure, such as root systems, leaf shapes, and water-storing tissues, physiological processes such as water and nutrient uptake, photosynthesis and transpiration, and life cycles such as timing of flowering and seed production. Annuals are plants that complete their life cycle within 1 growing season, producing seeds and then dying. Perennials are plants that live for multiple years, regrowing from the same roots or base each year. Biennials have a 2-year life cycle, typically producing leaves and stems in the first year and flowers and seeds in the second year before dying. Plants have evolved various adaptations to attract pollinators, such as brightly coloured flowers, sweet nectar, pleasant fragrances, and specialised structures to accommodate specific pollinators like bees, butterflies, birds, and bats. Some plants also mimic the appearance or scent of female insects to attract male pollinators. Monocots and dicots are 2 categories of flowering plants based on the number of seed leaves present in their embryos. Monocots have 1 seed leaf, parallel leaf veins, flower parts in multiples of 3, and scattered vascular bundles in their stems. Dicots have 2 seed leaves, net-like leaf veins, flower parts in multiples of 4 or 5, and vascular bundles arranged in a ring in their stems. The main parts of a tree include the roots for anchorage and nutrient absorption, trunk for support and water transport, branches for leaf exposure and photosynthesis, leaves for photosynthesis, and reproductive structures such as flowers and seeds. Trees benefit the environment in numerous ways. They absorb carbon dioxide and release oxygen through photosynthesis, improve air quality by trapping pollutants, provide habitats for wildlife, stabilize soil and prevent erosion, regulate water cycles by absorbing and releasing water, and offer aesthetic and recreational value. Plants have developed various defenses against herbivores, including physical barriers such as thorns, spines, and tough leaves, chemical defenses such as toxic compounds and deterrents, and symbiotic relationships with predators or parasites of herbivores. Fungi play crucial roles in ecosystems as decomposers, breaking down organic matter and recycling nutrients, forming symbiotic relationships with plants to aid in nutrient absorption and serving as food sources for other organisms. They also contribute to the production of antibiotics, enzymes, and other valuable compounds. Fungi obtain nutrients through extracellular digestion, secreting enzymes to break down complex organic compounds outside their cells and then absorbing the resulting simpler molecules. Some fungi are saprophytic, decomposing dead organic matter, while others are parasitic or mutualistic, forming symbiotic relationships with other organisms. And the main types of fungi include yeasts, molds, mushrooms, and lichens. Yeasts are single-celled fungi that reproduce asexually by budding. Molds are multicellular fungi with branching filaments called hyphae. Mushrooms are fungi with a fruiting body that produces spores for reproduction. Lichens are symbiotic associations between fungi and algae or cyanobacteria. Fungi differ from molds in that molds are a type of fungus characterized by their filamentous growth and ability to produce spores for reproduction. While molds can be harmful and cause food spoilage or health issues, many other fungi have beneficial roles in ecosystems. Fungi reproduce through both sexual and asexual means. Sexual reproduction involves the fusion of specialized reproductive cells called gametes from different mating types to form a zygote, which develops into a new organism. Asexual reproduction involves the production of spores through mitosis, which can then germinate and grow into new individuals. Bacteria play vital roles in ecosystems as decomposers, nutrient recyclers, nitrogen fixers, and symbionts. They are essential for nutrient cycling, soil fertility, plant growth, and the breakdown of organic matter. Some bacteria can cause diseases in plants and animals, but many others are beneficial or harmless. Bacteria are single-celled microorganisms that are among the simplest and most abundant life forms on Earth. They have a wide range of shapes, including spheres, rods, and spirals, and they can be found in various environments from soil and water to inside the human body. Bacteria lack a nucleus and other membrane-bound organelles found in eukaryotic cells. Bacteria reproduce through a process called binary fission, where a single bacterial cell divides into 2 identical daughter cells. This rapid reproduction allows bacteria to multiply rapidly under favorable conditions, leading to exponential population growth. Viruses and bacteria are both microscopic pathogens, but they differ significantly in structure, behavior, and reproduction. Bacteria are living organisms composed of cells with DNA or RNA genomes, while viruses are non-living particles consisting of genetic material enclosed in a protein coat called a capsid. Bacteria can reproduce independently, while viruses require a host cell to replicate. Viruses infect cells by attaching to specific receptors on the surface of host cells and injecting their genetic material into the cell. Once inside, the viral genetic material takes over the host cell's machinery to produce new viral particles, which can then infect other cells. Vaccines are biological preparations that stimulate the immune system to produce immunity to a specific pathogen, such as a virus or bacterium, without causing the disease itself. Vaccines typically contain weakened or inactivated forms of the pathogen or its components, which trigger an immune response, including the production of antibodies that provide protection against future infections. An epidemic is the rapid spread of a disease within a specific geographic area or population, affecting a larger number of people than expected. A pandemic, on the other hand, is an epidemic that has spread across multiple countries or continents, affecting a significant proportion of the global population. Antibiotics are medications used to treat bacterial infections by killing or inhibiting the growth of bacteria. They work by targeting specific bacterial structures or processes such as cell walls, protein synthesis, or DNA replication, while sparing human cells. However, overuse and misuse of antibiotics have led to the emergence of antibiotic-resistant bacteria, which can survive and multiply despite antibiotic treatment. Antibiotic resistance occurs when bacteria develop the ability to withstand the effects of antibiotics, rendering the drugs ineffective. This resistance can arise through genetic mutations or the acquisition of resistance genes from other bacteria. Antibiotic resistance is a significant public health concern, as it can lead to treatment failures, prolonged illness, and increased healthcare costs. Some bacteria have beneficial roles in various ecosystems and human activities. For example, certain bacteria help decompose organic matter, fix nitrogen in the soil, produce antibiotics and other useful compounds, and ferment foods and beverages. Bacteria also play essential roles in the human microbiome, aiding in digestion, immune function, and vitamin production. The human microbiome refers to the diverse community of microorganisms, including bacteria, viruses, fungi, and archaea that inhabit the human body, particularly the skin, mouth, gut, and other mucosal surfaces. These microbes play crucial roles in maintaining health and homeostasis, influencing various physiological processes and contributing to disease resistance. Bacteria help with digestion by breaking down complex carbohydrates, proteins, and fats in food into simpler molecules that can be absorbed and utilized by the body. In the gut, beneficial bacteria produce enzymes that aid in digestion, synthesize vitamins such as vitamin K and certain B vitamins, and compete with harmful pathogens for nutrients and space, thereby promoting gut health. While many bacteria are harmless or beneficial, some can cause diseases in humans and other organisms. Examples of harmful bacteria include pathogens such as Escherichia coli, Salmonella, Streptococcus, Staphylococcus, and Mycobacterium tuberculosis, which can cause a range of illnesses from mild infections to life-threatening diseases. Prokaryotic cells, such as those found in bacteria and archaea, lack a distinct nucleus and membrane-bound organelles. In contrast, eukaryotic cells, found in plants, animals, fungi, and protists, have a true nucleus containing the cell's genetic material and membrane-bound organelles, such as mitochondria, chloroplasts, the endoplasmic reticulum, and the Golgi apparatus. The cell membrane, also known as the plasma membrane, is a bilayer with embedded proteins that surrounds the cell, separating its internal environment from the external environment. The cell membrane controls the movement of substances into and out of the cell, allowing nutrients to enter and waste products to exit while maintaining cellular homeostasis. It also plays a crucial role in cell signaling, cell recognition, and adhesion. Diffusion and osmosis are both processes by which substances move across a membrane, but they differ in how they occur. Diffusion involves the movement of molecules from an area of higher concentration to an area of lower concentration, driven by the random motion of particles. Osmosis, on the other hand, specifically refers to the movement of water molecules across a selectively permeable membrane from an area of lower solute concentration to an area of higher solute concentration, typically to equalize the concentration of solutes on both sides of the membrane. This reduction in chromosome number ensures that when gametes combine during fertilization, the resulting zygote will have the correct number of chromosomes. Cells maintain homeostasis or internal balance by regulating various physiological processes such as temperature, pH, and nutrient levels to ensure optimal conditions for cellular function. This is achieved through mechanisms such as feedback loops, where changes in internal conditions trigger responses to restore equilibrium, and membrane transport processes, which control the movement of substances into and out of the cell. The Golgi apparatus, or Golgi complex, is a cellular organelle involved in processing, sorting, and packaging proteins and lipids for transport to their final destinations within the cell or for secretion outside the cell. It consists of a series of flattened membrane-bound sacs called cisternae, where proteins and lipids are modified by enzymes and then packaged into vesicles for transport. Plant and animal cells share many similarities, but also have several distinct differences. Plant cells have a rigid cell wall made of cellulose, chloroplasts for photosynthesis, and large central vacuoles for storing water and nutrients, while animal cells lack these structures. Animal cells typically have specialized structures called lysosomes for breaking down waste materials, which are less common in plant cells. Chloroplasts are organelles found in plant cells, and some algae that are responsible for photosynthesis, the process by which light energy is converted into chemical energy in the form of glucose. Chloroplasts contain chlorophyll, a green pigment that captures light energy, as well as other enzymes and molecules involved in the photosynthetic process. Active transport and passive transport are two mechanisms by which substances move across cell membranes. In active transport, molecules move against their concentration gradient, from an area of lower concentration to an area of higher concentration, requiring energy in the form of AT and the assistance of transport proteins. In passive transport, molecules move down their concentration gradient, from an area of higher concentration to an area of lower concentration, without the input of energy. Cells contain various organelles, each with specific functions. The nucleus houses the cell's genetic material and controls gene expression. The mitochondria are responsible for producing energy through cellular respiration. The endoplasmic reticulum is involved in protein synthesis and lipid metabolism. The Golgi apparatus processes and packages proteins for transport. The lysosomes contain enzymes for digesting waste materials. The cytoskeleton provides structural support and facilitates cell movement. The cell membrane, also known as the plasma membrane, surrounds the cell and regulates the passage of molecules in and out of the cell. It consists of a lipid bilayer embedded with proteins that control the transport of ions and molecules across the membrane. This selective permeability allows cells to maintain internal conditions necessary for survival while interacting with their environment. Cytoplasm is the gel-like substance found inside cells that contains organelles, cytoskeleton, and various molecules necessary for cellular processes. It serves as a medium for biochemical reactions and provides structural support to organelles. The nucleus is the control center of the cell, containing the cell's DNA, organized into chromosomes. It regulates gene expression, DNA replication, and cell division. The nucleolus located within the nucleus is responsible for producing ribosomes. Mitosis is the process of cell division that produces two identical daughter cells with the same genetic material as the parent cell. It ensures genetic stability by accurately distributing chromosomes to each daughter cell during cell division. Meiosis is a specialized type of cell division that produces gametes, such as sperm and eggs, for sexual reproduction. It reduces the chromosome number by half, ensuring genetic diversity in offspring. Cell differentiation is the process by which cells become specialized to perform specific functions. It involves changes in gene expression that lead to the development of distinct cell types with unique structures and functions. Stem cells are undifferentiated cells with the potential to develop into various cell types. They hold promise for medical research and therapy due to their ability to regenerate damaged tissues and organs. Cancer is a disease characterized by uncontrolled cell growth and division. It results from mutations in genes that regulate cell proliferation, apoptosis, and DNA. These mutations disrupt normal cellular processes, leading to the formation of tumors and metastasis. Mutations can arise from various sources, including exposure to radiation, chemicals, and viruses, as well as errors in DNA replication and repair. They contribute to genetic diversity within populations and can have both beneficial and harmful effects on organisms. DNA sequencing is the process of determining the precise order of nucleotides in a DNA molecule. It has revolutionized genetics by providing insights into the structure, function, and evolution of genomes. There are several methods used for DNA sequencing, including Sanger sequencing, next-generation sequencing, and single-molecule sequencing. These techniques differ in their approach to DNA amplification, sequencing chemistry, and data analysis. Gene editing is a technology that allows scientists to modify DNA sequences within an organism's genome. It holds promise for treating genetic diseases, engineering crops with desirable traits, and advancing basic research in genetics and molecular biology. Genetic engineering is the process of manipulating the genetic material of organisms to produce desired traits. It is used in agriculture to improve crop yield, resistance to pests and diseases, and nutritional content. In medicine, genetic engineering is used to produce recombinant proteins, gene therapies, and genetically modified organisms for research and therapeutic purposes. Ethical considerations surrounding genetic manipulation revolve around questions of consent, equity, safety, and unintended consequences. Scientists and ethicists debate the ethical implications of genetically modifying organisms, including humans, for purposes such as disease prevention, enhancement, and altering traits. Concerns also arise regarding potential misuse of genetic technologies, inequitable access to genetic interventions, and the long-term impact on ecosystems and biodiversity. Inheritance patterns refer to how traits are passed from parents to offspring. There are various types of inheritance, including autosomal dominant, autosomal recessive, X-linked dominant, X-linked recessive, and mitochondrial inheritance. These patterns determine how genetic traits are expressed in individuals and can influence the likelihood of certain traits appearing in future generations. Mendelian genetics, named after Gregor Mendel, explores the inheritance of traits controlled by single genes with distinct dominant and recessive alleles. Mendel's experiments with pea plants in the 19th century revealed patterns of inheritance that laid the foundation for modern genetics. He discovered principles such as segregation and independent assortment, which describe how alleles are distributed during gametake formation and inheritance. Dominant alleles mask the expression of recessive alleles when present in a heterozygous individual. In genetic crosses between organisms with different alleles, dominant traits are typically observed in the offspring, while recessive traits may only appear when both alleles are recessive. This interaction between alleles determines the phenotypic expression of traits in offspring. Incomplete dominance occurs when neither allele is dominant, resulting in a blending of traits in heterozygous individuals. This leads to an intermediate phenotype that is distinct from either homozygous parent. An example is snapdragon flower color, where red and white alleles produce pink flowers in heterozygous plants. Codominance occurs when both alleles contribute equally to the phenotype, resulting in the simultaneous expression of both traits in heterozygous individuals. An example is the AB blood type in humans, where both A and B alleles are expressed on the surface of red blood cells. Sex-linked traits are controlled by genes located on sex chromosomes, typically the X chromosome. These traits are inherited differently in males and females due to differences in sex chromosome inheritance. In males, recessive X-linked traits are expressed more often because they only have one X chromosome, while females must inherit 2 copies of the recessive allele to express the trait. Polygenic inheritance occurs when a trait is influenced by multiple genes, each contributing incrementally to the phenotype. This type of inheritance results in continuous variation within a population and contributes to traits such as height, skin color, and intelligence. Pedigrees are diagrams that show the inheritance of traits within a family over multiple generations. They are used by geneticists to analyze patterns of inheritance, identify carriers of genetic disorders, and assess the risk of passing on certain traits to offspring. Genetic counselling is a service provided to individuals and families to help them understand the genetic basis of inherited conditions, assess their risk of developing or passing on genetic disorders, and make informed decisions about testing, treatment, and family planning. Genetic counsellors provide information, support, and guidance to individuals seeking to navigate the complex landscape of genetics and genomics. Evolutionary biology is the study of how species evolve and diversify over time. It explores the processes of natural selection, genetic drift, gene flow, mutation, and speciation that drive changes in populations and lead to the diversity of life forms on Earth. Natural selection is the process by which organisms with traits that enhance their survival and reproductive success are more likely to pass on those traits to future generations. Over time, natural selection leads to the adaptation of populations to their environments and the emergence of new species. Mutations are changes in the DNA sequence that can result from errors in DNA replication, exposure to mutagens, or other factors. Mutations introduce genetic variation within populations, which is the raw material for evolution. Genetic drift is the random fluctuation of allele frequencies in small populations due to chance events. It can lead to the loss of genetic diversity over time and may result in the fixation of certain alleles or the loss of others. Gene flow is the movement of genes between populations through migration or interbreeding. It introduces new alleles into populations, increasing genetic diversity and promoting adaptation to changing environments. Gene flow can also reduce genetic differences between populations and maintain genetic homogeneity within species. Speciation is the process by which new species arise from existing ones. It occurs when populations of a species become reproductively isolated from each other, preventing gene flow and allowing genetic divergence to accumulate over time. There are several mechanisms by which speciation can occur, including allopatric speciation, sympatric speciation, and parapatric speciation, each involving different forms of geographic or reproductive isolation. Adaptive radiation is a process in which a single ancestral species diversifies into a wide variety of ecological niches, leading to the rapid evolution of multiple new species. It often occurs in response to the colonization of new habitats or the availability of new resources, resulting in the evolution of diverse adaptations among related species. Convergent evolution is the independent evolution of similar traits in unrelated species in response to similar selective pressures or environmental conditions. It results in organisms that are not closely related developing analogous structures or behaviours due to shared ecological challenges. Coevolution is the reciprocal evolutionary change between two or more interacting species. It occurs when species exert selective pressures on each other, leading to adaptations that benefit both parties. Examples of coevolution include predator-prey relationships, mutualistic symbioses, and host-parasite interactions. The fossil record is the collection of preserved remains or traces of ancient organisms found in sedimentary rock layers. It provides evidence for evolution by documenting the diversity of life over geological time and the transitions between different forms. Fossils help scientists reconstruct the evolutionary history of organisms and understand the patterns and processes of evolution. Comparative anatomy is the study of the similarities and differences in the anatomy of different species. It reveals evolutionary relationships by identifying homologous structures, traits inherited from a common ancestor, and analysing their variations among related organisms. Comparative anatomy provides insights into the evolutionary changes that have occurred over time, and the anatomical adaptations of organisms to different environments. Molecular biology is the study of biological molecules, particularly deoxyribonucleic acid and proteins and their functions in living organisms. It provides insights into evolutionary history by comparing the genetic sequences and molecular structures of different species. Molecular data help scientists reconstruct phylogenetic relationships, estimate divergence, times between species, and infer evolutionary processes such as genetic drift and natural selection. Biogeography is the study of the distribution of organisms across geographical space and through geological time. It supports the theory of evolution by documenting patterns of species distribution, migration, and extinction. Biogeographic patterns reflect the historical processes of speciation, dispersal, and vicariance that have shaped the distribution of life on Earth. Embryology is the study of the development embryos and how they grow and differentiate into mature organisms. It provides evidence for evolutionary relationships by comparing the embryonic development of different species. Embryological similarities among related organisms reflect their common ancestry and evolutionary history, revealing patterns of evolutionary change and adaptation. The theory of punctuated equilibrium proposes that evolutionary change occurs in rapid bursts of speciation, separated by long periods of stasis, during which species remain relatively unchanged. It contrasts with the model of gradualism, which suggests that evolutionary change occurs gradually and continuously over time. Punctuated equilibrium emphasizes the role of speciation events in driving evolutionary patterns and the importance of environmental factors in shaping the pace of evolution. Extinction is the permanent loss of a species or group of organisms. It plays a significant role in shaping evolutionary patterns by removing lineages from the tree of life and creating opportunities for new species to evolve and diversify. Mass extinctions in particular have had profound effects on the course of evolution, leading to the rise of new species and the restructuring of ecological communities. Homologous structures are anatomical features that have a common evolutionary origin, but may have different functions in different organisms. They provide evidence for common descent and shared ancestry among related species. Examples include the pentadactyl limb of vertebrates and the forelimbs of mammals, which have similar bone structure despite serving different functions. Vestigial structures are anatomical features that have lost their original function over the course of evolution, but are retained in organisms as remnants of their evolutionary history. They provide evidence for evolutionary change and adaptation. Examples include the human appendix, wisdom teeth, and hind limb bones in whales, which are reduced or non-functional in modern organisms but were functional in their evolutionary ancestors. The molecular clock hypothesis proposes that the rate of evolutionary change in deoxyribonucleic acid sequences is constant over time and can be used to estimate the divergence times between species. It relies on the assumption that mutations accumulate at a constant rate in the genome, allowing scientists to calibrate the molecular clock using known divergence events and apply it to estimate the timing of evolutionary events that occurred in the past. The Hardy-Weinberg equilibrium is a principle in population genetics that describes the theoretical distribution of allele frequencies in a population that is not evolving. It states that under certain conditions, such as random mating, no migration, no mutation, and no selection, and infinite population size, allele frequencies will remain constant from generation to generation. Deviations from Hardy-Weinberg equilibrium indicate that evolutionary processes such as genetic drift, natural selection, or gene flow acting on the population. The founder effect occurs when a small group of individuals from a larger population establishes a new population in a different geographic area due to the limited genetic diversity of the founding population. The new population may have reduced genetic variation compared to the original population. Over time, this can lead to genetic differences between the two populations, as certain alleles become more common or rare in the founder population due to chance. The bottleneck effect occurs when a population undergoes a drastic reduction in size, often due to natural disasters, habitat destruction, or human activities. As a result of the population bottleneck, genetic diversity is significantly reduced, as only a subset of the original genetic variation is represented in the surviving individuals. This loss of genetic diversity can have long-term consequences, as the population may become more vulnerable to environmental changes and genetic disorders. The biological species concept defines a species as a group of organisms that can interbreed and produce fertile offspring under natural conditions. According to this concept, species boundaries are determined by reproductive compatibility rather than physical or ecological characteristics. Prezygotic reproductive barriers prevent individuals of different species from mating or fertilizing each other's eggs, while postzygotic barriers prevent hybrid offspring from developing into viable, fertile adults. Allopatric speciation occurs when populations of the same species become geographically isolated from each other, leading to reproductive isolation and the evolution of distinct species. Geographic barriers such as mountains, rivers, or islands can prevent gene flow between populations, allowing genetic divergence to accumulate over time. Sympatric speciation, on the other hand, occurs when speciation occurs in populations that live in the same geographic area. This can happen through mechanisms such as disruptive selection, polyploidy, or ecological specialization. Sexual selection is a process in which individuals with certain traits are more likely to mate and reproduce than others, leading to the evolution of exaggerated traits such as elaborate ornaments or displays. Sexual selection can contribute to speciation by promoting reproductive isolation between populations with different mating preferences or behaviours. For example, if females prefer males with specific mating displays. Males from different populations may evolve different displays, leading to reproductive isolation and eventually speciation. Reproductive isolation refers to the barriers that prevent gene flow between populations, leading to the evolution of distinct species. These barriers can be prezygotic, occurring before mating or fertilization, or postzygotic, occurring after mating or fertilization. Prezygotic barriers include mechanisms such as temporal isolation, behavioral isolation, and mechanical isolation, while postzygotic barriers include reduced hybrid viability or fertility. Ring species are populations that are connected by a ring-like distribution around a geographic barrier, such as a mountain range or body of water. Each population can interbreed with its neighbouring populations, but the populations at the ends of the ring are reproductively isolated and unable to interbreed. Ring species provide insights into the process of speciation by illustrating how geographic isolation and reproductive barriers can lead to the evolution of new species. Evolutionary developmental biology is a field of biology that studies how changes in developmental processes contribute to evolutionary change. Evolutionary developmental investigates the genetic and molecular mechanisms underlying the development of organisms, and how changes in these mechanisms can lead to morphological diversity and evolutionary novelties. Homitic genes are master regulatory genes that control the development of body structures by determining the identity of different body segments. Changes in homeotic gene expression can lead to the evolution of new body structures or alterations in existing structures. Modularity is a concept in evolutionary biology that refers to the organization of biological systems into discrete, functionally independent units or modules. Modular organization allows organisms to evolve new traits or behaviors without disrupting existing structures or functions. Changes in one module may have limited effects on other modules, providing flexibility and evolvability. Heterochrony is a process in which changes in the timing of developmental events lead to differences in the timing of trait expression. Heterochronic changes can result in the acceleration or delay of developmental processes, leading to morphological differences between species. Evolutionary novelties are newly evolved traits or features that are unique to a particular lineage or species. They arise through genetic changes such as mutations or gene duplications that produce novel functions or structures. Evolutionary novelties can contribute to adaptive radiation, speciation, and ecological diversification by providing organisms with new capabilities or adaptations. Mutations are changes in the DNA sequence of an organism's genome that can result in phenotypic diversity and contribute to evolutionary change. Evolutionary constraints are limitations imposed by historical, genetic, developmental, or ecological factors that restrict the range of possible phenotypes that organisms can evolve. These constraints can arise from genetic pleiotropy, where a single gene influences multiple traits, or from trade-offs between different traits that impose competing selective pressures. For example, the architecture of the vertebrate eye is constrained by its evolutionary history, resulting in suboptimal structures such as the blind spot. Developmental plasticity refers to the ability of organisms to produce different phenotypes in response to environmental cues during development. This plasticity can influence evolutionary trajectories by allowing organisms to adapt to changing environmental conditions without genetic changes. For example, plants may adjust their growth patterns in response to changes in light or nutrient availability, leading to the evolution of different morphologies in different environments. Evolutionary convergence occurs when unrelated species evolve similar traits or adaptations independently in response to similar selective pressures. This phenomenon is driven by natural selection acting on similar ecological niches or environmental conditions. For example, the streamlined body shapes of dolphins. And sharks are convergent adaptations for efficient swimming in aquatic environments, despite their different evolutionary histories. Evolutionary divergence occurs when populations of the same species evolve different traits or adaptations in response to different selective pressures or environmental conditions. This divergence can lead to the formation of new species over time. The factors such as geographic isolation, ecological specialization, or reproductive isolation can contribute to evolutionary divergence by reducing gene flow between populations. The Red Queen hypothesis proposes that organisms must continually evolve and adapt in order to survive and reproduce in changing environments. The name is inspired by Lewis Carroll's character, who famously said, "Now here, you see, it takes all the running you can do to keep in the same place." The hypothesis suggests that organisms must constantly evolve to outpace co-evolving predators, parasites, or competitors, maintaining an evolutionary arms race for survival. Coevolution is the reciprocal evolutionary change that occurs between interacting species as a result of selective pressures imposed by their ecological interactions. This process can lead to mutualistic relationships, such as the coevolution between flowering plants and their pollinators, or antagonistic relationships, such as the coevolution between hosts and their parasites or predators. Genetic drift is a random process that can lead to changes in allele frequencies within populations over time. This process is particularly influential in small populations, where chance events can have a disproportionate impact on allele frequencies. Genetic drift can lead to the fixation or loss of alleles, reducing genetic variation within populations and increasing genetic differentiation between populations. The neutral theory of molecular evolution proposes that the majority of genetic variation within and between species is caused by random genetic drift rather than natural selection. According to this theory, most mutations are selectively neutral or nearly neutral, meaning they have little or no effect on an organism's fitness. Therefore, genetic variation accumulates over time primarily due to random processes rather than adaptive evolution. The Baldwin effect suggests that learned behaviours or phenotypic plasticity can influence the direction and rate of evolution by providing a bridge between environmental change and genetic adaptation. In other words, behaviours or traits that are initially acquired through learning or development can become genetically encoded over time through natural selection. For example, the ability to digest novel foods or tolerate new environmental conditions may initially be learned behaviours, but if they provide a fitness advantage, they may become genetically fixed in a population. Epigenetics refers to changes in gene expression that are not caused by alterations in the deoxyribonucleic acid sequence itself. These changes can be influenced by environmental factors such as diet, stress, or exposure to toxins, as well as by developmental processes. Epigenetic modifications, such as deoxyribonucleic acid methylation or histone modification, can alter gene expression patterns and contribute to phenotypic variation and adaptation. The extended evolutionary synthesis is a framework that expands upon traditional evolutionary theory by integrating insights from fields such as developmental biology, ecology, and epigenetics. This approach emphasizes the importance of non-genetic inheritance mechanisms, such as epigenetic modifications or cultural transmission, in shaping evolutionary trajectories. By considering the dynamic interactions between genes, development, and the environment, the extended evolutionary synthesis provides a more comprehensive understanding of evolutionary processes. Evolutionary mismatch refers to situations where modern environments and lifestyles differ significantly from those in which humans evolved, leading to a misalignment between our biological adaptations and our current circumstances. This mismatch can contribute to various health problems in modern society. For example, our sedentary lifestyles and high-calorie diets can lead to obesity and metabolic disorders because our bodies are adapted for environments where food was scarce and physical activity was essential for survival. Many human diseases such as cancer, diabetes, and heart disease have evolutionary origins rooted in our evolutionary history. For instance, the genes that predispose individuals to these diseases may have provided advantages in the past, such as enhanced energy storage or resistance to infections, but they can become detrimental in modern environments with abundant food and sedentary lifestyles. Infectious diseases have played a significant role in shaping human evolution by exerting selective pressures on our immune systems. Throughout our evolutionary history, pathogens have posed constant threats to our survival, driving the evolution of immune defense mechanisms. However, the rapid spread of infectious diseases in densely populated modern societies can overwhelm these defenses, leading to outbreaks and pandemics. Humans have evolved numerous adaptations that reflect our evolutionary history, such as bipedal locomotion, dexterous hands, and large brains. These adaptations allowed our ancestors to thrive in diverse environments and exploit new resources, ultimately shaping our evolutionary trajectory. Aging is a complex biological process with evolutionary significance. While aging reduces an individual's reproductive fitness over time, it may have evolved because the benefits of maintaining youthfulness decline with age, and resources are better allocated to reproduction earlier in life. Additionally, some theories propose that aging may have evolved as a byproduct of other processes, such as deoxyribonucleic acid damage accumulation or antagonistic pleiotropy. Antagonistic pleiotropy occurs when a single gene influences multiple traits, some of which may have opposing effects on an organism's fitness. For example, genes that promote early reproductive success may also increase the risk of age-related diseases later in life. This trade-off between early reproductive success and long-term health may contribute to aging age-related diseases. Gene culture coevolution occurs when cultural practices or behaviours shape the genetic evolution of populations, and vice versa. For example, the development of agriculture led to changes in human diets, which in turn influenced the evolution of genes related to lactose tolerance or starch digestion. Altruism or selfless behaviour, that benefits others at a cost to oneself has evolutionary origins rooted in kind selection, reciprocal altruism, and group selection. By helping relatives or cooperating with non-relatives, individuals can increase the inclusive fitness of their genes or enhance the survival and reproduction of their social groups. Evolutionary arms races occur when species engage in reciprocal adaptations to gain advantages in competitive or antagonistic interactions. For example, the co-evolutionary dynamics between predators and prey, hosts and parasites, or competitors can lead to the escalation of traits or behaviours that confer a competitive advantage. Epigenetic inheritance refers to the transmission of epigenetic modifications from one generation to the next, potentially influencing gene expression and phenotypic traits. Environmental factors such as diet, stress, or exposure to toxins can induce epigenetic changes that may be passed on to offspring, shaping their developmental trajectories and physiological responses. Human activities such as urbanization, agriculture, and industrialization have caused rapid environmental changes that exert selective pressures on populations, driving evolutionary responses. For instance, the spray of antibiotic resistance in bacteria, or the adaptation of pest species to pesticides are examples of rapid evolution in response to human interventions. Symbiotic relationships between species, such as mutualism, commensalism, or parasitism, have evolved as a result of selective pressures to maximize fitness. These relationships can lead to co-evolutionary dynamics where interacting species influence each other's evolution over time. Social behaviours such as cooperation, reciprocity, and competition have evolutionary origins shaped by selective pressures to maximise reproductive success or inclusive fitness. These behaviours can be adaptive strategies for navigating social environments, forming alliances, or competing for resources. Kin selection refers to the evolutionary process by which organisms exhibit altruistic behaviours that benefit their relatives, often at a cost to themselves. This behaviour can be explained by the fact that relatives share a portion of their genes, so helping relatives enhances the chances of passing on shared genetic material. For example, bees in a hive work together for the benefit of the colony, as they are closely related and share genetic material. Convergent evolution occurs when unrelated species independently evolve similar traits or adaptations in response to similar environmental challenges. For instance, both bats and birds have evolved wings for flight, despite their different evolutionary lineages, as they both faced the challenge of airborne locomotion in their environments. Sexual dimorphism refers to differences in size, shape, or other physical characteristics between males and females of the same species. These differences often evolve as a result of sexual selection, where individuals compete for mates or are chosen by mates based on these traits. For example, in many bird species, members may have bright plumage or elaborate displays to attract mates, while females have more cryptic coloration to avoid predators while nesting. Evolutionary trade-offs occur when individuals face conflicting demands on limited resources, leading to compromises between different traits or functions. For instance, investing more resources in reproduction may come at the expense of survival or vice versa. An example of this trade-off can be seen in the life history strategies of organisms, where species that produce many offspring tend to have shorter lifespans compared to those that invest more in individual survival. Gene flow, or the movement of genes between populations, plays a crucial role in maintaining genetic diversity within populations. This occurs through migration, where individuals move between populations and interbreed, exchanging genetic material. Gene flow helps prevent populations from becoming genetically isolated and can introduce new allies into a population, increasing genetic variation. Evolutionary adaptations in response to changing climates are widespread across various species. For example, In response to warming temperatures, some species may shift their ranges toward higher latitudes or elevations, while others may adapt by changing their behaviours or physiology to cope with new environmental conditions. Human cognition, including abilities such as problem solving, social learning, and language, has evolved over millions of years. These cognitive abilities likely evolved as adaptations to the complex social and ecological challenges faced by our ancestors, such as navigating social hierarchies, cooperating in groups, and acquiring and transmitting knowledge. Evolutionary vestiges are traits or structures that have lost their original function over the course of evolution, but are retained in descendants. For example, the human appendix is thought to be a vestigial structure that once played a role in digesting tough plant material, but is now non-functional in most people. Biodiversity, or the variety of life on Earth, is essential for ecosystem functioning and resilience. It provides ecosystem services such as pollination, nutrient cycling, and disease regulation, and increases the stability of ecosystems in the face of environmental change. Evolutionary innovations are adaptations or traits that arise and spread throughout a population, leading to significant changes in biological diversity and ecological interactions. Examples include the evolution of flight in birds and insects, the development of flowers and angiosperms, and the evolution of jaws in vertebrates. Major transitions in biological complexity refer to key events in evolutionary history where groups of organisms underwent significant changes in their organization, behavior, or ecological interactions. Examples include the transition from single-celled organisms to multicellular life, the evolution of eusociality in insects, and the development of complex social behaviors in primates. Evolutionary convergence occurs when distantly related species independently evolve similar traits or adaptations in response to similar selective pressures. For example, dolphins and sharks have both evolved streamlined bodies and fins for efficient swimming, despite their different evolutionary lineages. The cooperation and altruism are common in social species and have evolved as strategies to enhance the survival and reproduction of individuals within groups. Examples include cooperative hunting in carnivores, cooperative breeding in birds, and reciprocal altruism in primates. Evolutionary strategies for avoiding predation and increasing survival include a wide range of adaptations such as camouflage, warning coloration, defensive behaviors, and physical defenses like spines or shells. These strategies evolve through natural selection as individuals with traits that enhance survival are more likely to pass on their genes to the next generation. Genetic diversity within populations provides resilience to environmental change by increasing the likelihood that some individuals will possess traits that allow them to survive and reproduce under new conditions. This genetic variation allows populations to adapt to changing environments through natural selection, reducing the risk of extinction. Evolutionary adaptations in response to novel environments are widespread across various organisms. For instance, the Galapagos finches develop different beak shapes and sizes to exploit different food sources on the islands, demonstrating how natural selection drives adaptations in response to environmental challenges. Phenotypic plasticity, or the ability of an organism to change its phenotype in response to environmental cues, allows individuals to cope with changes in their surroundings. Examples include changes in coloration or behavior in response to temperature or light conditions. Evolutionary innovations that lead to ecological dominance involve traits or behaviors that give certain species a competitive advantage in their ecosystems. Examples include the evolution of photosynthesis in plants, which allowed them to dominate terrestrial environments, and the development of jaws in early vertebrates, which facilitated predation and diversification. Evolutionary constraints limit the range of possible phenotypes that can evolve due to genetic, developmental, or ecological factors. Examples include developmental constraints that restrict the types of structures that can evolve, such as the constraints on limb structure in tetrapods imposed by their evolutionary history as fish. Evolutionary convergence at the molecular level refers to the independent evolution of similar genetic sequences or biochemical pathways in different lineages. Examples include the convergent evolution of genes involved in vision in vertebrates and cephalopods, despite their distant evolutionary relationships. The evolutionary history of major anatomical structures and physiological systems traces the gradual accumulation of adaptations over millions of years. Examples include the evolution of vertebrate limbs from fish fins and the development of complex nervous systems in bilaterally symmetric animals. Evolutionary innovations that transform ecosystems and landscapes often involve key adaptations or ecological interactions that have cascading effects on the environment. Examples include the evolution of flowering plants, which transform terrestrial ecosystems by diversifying habitats and supporting new forms of life, and the colonization of land by tetrapods, which altered terrestrial landscapes and ecosystems. Evolutionary trade-offs play a crucial role in determining how organisms allocate limited resources among competing needs such as growth, reproduction, and survival. For example, investing more energy in reproduction may come at the expense of growth or immune function, highlighting the trade-offs organisms face in optimizing their fitness. Examples of evolutionary adaptations that enhance reproductive success include traits such as bright plumage in birds, elaborate courtship displays in insects, and efficient seed dispersal mechanisms in plants. These adaptations increase an individual's chances of attracting mates, successfully reproducing, and passing on their genes to the next generation. Ecological interactions such as competition, predation, and mutualism drive evolutionary change by selecting for traits that confer advantages in specific ecological context. For example, competition between species for limited resources can drive the evolution of resource partitioning and niche specialization, while mutualistic relationships between species can lead to coevolution and the development of interdependent traits. Evolutionary transitions between different modes of reproduction, such as sexual and asexual reproduction, involve shifts in reproductive strategies that can have profound effects on genetic diversity and population dynamics. For example, the transition from sexual reproduction to asexual reproduction in certain organisms can lead to the rapid proliferation of clones, but may also increase vulnerability to parasites and environmental fluctuations. Phenotypic variation within populations is the raw material upon which natural selection acts, driving evolutionary change over time. Examples include variations in beak size and shape among Darwin's finches, which have been shaped by differences in food availability and feeding behaviours on the Galapagos Islands. Genetic diversity within populations provides resilience to environmental disturbances by increasing the likelihood that some individuals will possess traits that confer resistance or tolerance to changing conditions. Examples include variations in disease resistance genes that protect certain individuals from pathogens, or variations in drought tolerance genes that enable plants to survive in arid environments. As we conclude today's exploration of biology, we hope you've gained a deeper appreciation for the diversity and complexity of life on Earth. Don't forget to subscribe to Loops and Loops for more captivating content on biology, ecology, and the wonders of the natural world. Let's continue to loop in new knowledge, nurture our curiosity, and deepen our understanding of the biological systems that shape our world. Until next time, keep those loops tight, keep exploring, and keep marveling at the wonders of biology. Thank you for joining us on this enlightening journey.